Hello. I want to thank the organizers for the opportunity to present about the regulatory aspects and considerations for high content diagnostic tests. I want to also thank all of you who joined. To start with some housekeeping items, if you do have any questions and if they come up during the presentation, just press that little Q&A button that's on your left, I think, bottom left, and please feel free to submit them during the talk, and I will try to address them as soon as the presentation is completed. As a way of introduction, my name is Zivana Tezak. I'm working in the Office of In Vitro Diagnostics and Radiological Health, which is OIR for short. This is the FDA's office that reviews and approves for marketing diagnostic tests. I will lay out some of the FDA's current thoughts on evaluation of high content tests, and I will be using some specific examples and mainly focusing on next generation sequencing, which has been a topic of many talks during this conference so far from what I've seen. And I'm hoping that this will give you an overview of the high content test policies we are working on at FDA. I do have to disclaim it, since there are no FDA cleared or approved next-gen sequencing tests at this point in time. Although very much in development, what I am presenting today is still preliminary, and there is no finalized FDA policy for evaluation and regulation of these tests just yet. We do encourage anybody with interest in submitting this type of novel clinical diagnostic tests or platforms to come in with a pre-submission to our office. So we can discuss your particular test and platform, and you can discuss it with the FDA team that will actually review it, and you can get the most current advice. My plan in this presentation is to give you a high-level overview of FDA in vitro diagnostic regulation, some kind of FDA 101, if you like. And then I will get down to some specifics applicable to high content and actually specifically NGS tests. I will compare and contrast some of the main points in FDA review for in vitro diagnostic tests in general, and then with the questions that are specifically related to evaluation of NGS tests. So starting with this FDA 101, what you see on this slide is the regulation, which is a part of the law, and it's defining FDA's jurisdiction over regulating in vitro diagnostic tests. Now, diagnostic tests in the US are regulating as a device, and they're a part, so the regulations for diagnostic tests are part of device regulations. If you're looking at this regulation, it encompasses different types of, you can put under quote, really diagnostic testing, because it's talking about the cure and mitigation and prevention of disease. So then on this slide are listed parts of regulation that's telling FDA how we should look at IVDs. And this is actually the same regulations as for devices in general. So we are supposed to evaluate safety and effectiveness of any new test. If you look at these definitions of both safety and effectiveness, you can see that they're pretty broad. So FDA has some freedom within these to establish the best policies and requirements for a particular device or particular test type. So as I'm sure a lot of people in the audience are well aware, there are a number of different ways one can look at the genomic tests in particular. So here I kind of categorize these tests in two very broad categories if you look at these two big bullet points. I'm categorizing them in single analyte tests, for example, a simple test for just one gene, or you can have multiplex tests or multiple marker tests, which are testing either a number of genes or a number of mutations in one gene, or a number of microorganisms. We just had the approval, but that was mass spec, not DNA testing, of I think 137 microorganisms. So you can think of next-gen sequencing as the ultimate multiplex testing, really. So what's listed also in this slide are arrays. And as an example, you can think of array-based copy number analysis. This is a common technology for copy number variation, or CNV detection. American College of Medical Genetics, or ACMG, recommends array CGH as a first-line diagnostics for abnormalities such as developmental delay, dysmorphic features, congenital anomalies, and so on. So looking at this slide, there are a number of FDA cleared or approved tests for both of these categories of genetic tests, so both single and multiple marker. And I listed some example claims in this slide. We do have a lot of experience with regulating these types of tests. Okay, so let's focus on high density, high content tests, and let's focus in particular on NGS sequencing based tests. FDA has cleared a number of multiplex tests, as I mentioned before, so that's not that new. So how are these NGS based tests different from other genetic or genomic tests that have been FDA cleared so far? Well, first, these technologies have been developing and changing extremely rapidly in the last few years. I'm sure I don't have to tell people who are listening that. If you're looking at the whole system, from getting a clinical sample through preparing and testing it, software for analyzing data, reporting back results, different parts of the system are manufactured by different manufacturers, and manufacturing them may require very different expertise. And then, more importantly, with genomic sequencing, unlike with multiplex tests where you have predefined variants or predefined microorganisms or analytes you're looking at, and again, we have FDA approved tests that test either genomic tests, talking about genomic tests, 60 plus possible mutations in one gene or 20 plus different microorganisms. So tests like sequencing, unlike those, will provide unlimited results. And some or many of these may have never been seen before, so these results. So how can you assess the clinical significance of these results? And then additionally, how can you evaluate analytical performance of all different possible outputs and understand that the results you will report are analytically valid? So we do like to put the issues we need to address up for public discussion with the experts in the specific field. And seems we have been doing a lot of these meetings and public discussions, and more lately. To start addressing some of the questions related to next-gen sequencing, we held a public meeting about 2 years ago, a little over 2 years ago, in summer of 2011, and there is a link to all the information related to that meeting at the bottom of this slide in blue. It's a little small. This meeting brought together many of the pioneers and top experts in the NGS field at that time, 2 years ago, including academics, governments, manufacturers, clinical labs, professional organizations, etc. In short, we asked, FDA has asked a lot of questions, and we put forth some suggestions for evaluation. And what we received back were mainly more questions. Again, that was 2 years ago. Not too many solutions on how to evaluate the performance of these tests. But since then, over the last 2 years, many of the answers to the questions that we have discussed at that meeting have been slowly falling into place. And many of the presentations actually today and yesterday were addressing some of those. So I will present some of these solutions in the course of this talk. So on this next slide, I'm listing for you the main parts which FDA review in order to understand the performance of a genomic test. And I will briefly cover some of these bullet points in the next few slides, but I will not go through all of them. At the bottom, I'm providing a link to the web page where we are listing all FDA approved or cleared nucleic acid-based tests. And from that link, you can pick the test that you may be interested in, and you can go to the pages for any individual approved test, and you can see there the summary of the test performance. So for every diagnostic test that is FDA approved or FDA cleared, we publish the summary of the information we reviewed. Again, it's just a summary. It's not all the line data and not all the raw data, not all the data that we reviewed, but the summarized information. And this is very transparent information. You can see the performance of the test. So you can see what works, what doesn't work, and what were the studies that were done. Okay, so I listed different parts of the submission on the previous slide, and I want to start with the intended use. So when FDA reviews tests, we start by looking at what is test going to be used for, what are the claims. And those claims may be very general, or they may be very specific, as in this particular example. So this specific example, this is intended use for the first FDA approved test in so-called IVD-MIA category, which stands for in vitro diagnostic multivariate index assay. It's kind of a retired category right now, but basically this is the test that measures multiple analytes and puts together the results in a multi-marker signature. That would be kind of a colloquial definition. The main parts of the intended use of the test are, as you can see in these blue bubbles, the analyte tested, the intended use population to be tested, which is very important and many times forgotten, and what are the indications for use. And then, so the types of studies to evaluate certain tests will all depend on the claim in the intended use, and it would be somewhat dependent on the technology that test is run on. So what would you want to know about the diagnostic test performance? It's pretty simple, really, and straightforward. You want to know that your test analytically measures what you think it does, and that it does it accurately. And you also want to know that your test results correlates with the clinical condition you're testing. So this next slide is listing analytical studies that we normally evaluate in order to understand the performance of a new test. And the studies listed here are not limited to any type of in vitro diagnostic tests. They're general studies for evaluation of any IVD. Where the specifics come into play is when you're looking at details of each of these studies as they relate to a particular test. So in the next few slides, I will highlight some of the performance evaluation parts that may be challenging or different for next-gen sequencing from the way we have been traditionally evaluating performance of the diagnostic test. So you can use the link in a few slides ago in slide 8 to see what performance studies need to be submitted to FDA in order for specific tests to be cleared or approved and you can see all these various studies in device summaries. So I will not go into those details here, but again, I will just try to highlight what's different for NGS tests, or what's specific for them. So starting with non-NGS genomic tests, right? So starting with accuracy. One of the analytical studies that may be very different from what we have been asking in the past is accuracy. To set it up, this slide highlights some of the parts of accuracy studies that are normally required for molecular diagnostic tests in general. So for FDA submissions, analytical accuracy of a new test is generally evaluated by comparing new test results to results of some accepted reference method. For example, for genetic tests, in many cases, bidirectional sequencing or Sanger sequencing has been regarded as the gold standard method to compare to. There are also cases where new test results have to be compared to a clinical truth because there is no good gold standard or reference method, or there is no good comparator. In order to assess accuracy, we generally require multiple real clinical samples for every output of the test. Now, why is that? Why multiple samples for every output? And I'm still talking, if you notice at the top, Still talking about genomic tests in general, not necessarily NGS or high content tests. So in this table, you can see results of a made-up multiplex test consisting of 23 alleles. Or you can imagine these as being different genes in cancer panel, or you can imagine these as being different viruses in the respiratory disease panel, for example. So the total positive agreement of the test, if you look at the top where it says total, is almost 99%. However, if you look down at allele 22, it has much lower accuracy than others, only about 67%. And if you are reporting this allele, or if this is a completely separate virus, for example, you need to know that. Showing just the accuracy of the whole test, which is 99%, would be misleading for this particular result, which is only 67%. So, of course, and I will elaborate a bit more later, we cannot require this kind of evaluation for next-gen sequencing-based tests in most cases. Many times you cannot even predict, let alone adequately evaluate every single output of the next-gen sequencing tests. So how can we assess then accuracy of NGS-based tests? A suggested approach is to select a subset of markers, such as the ones listed here in those three small bullet points. So analytically challenging markers from relevant disorders, redundant sequences, spanning the genome, etc. And then if we evaluate the performance of these, in order to, we can infer the performance of the whole platform from their performance. Of course, this opens many other questions. And some are listed on this slide, including what would be an appropriate comparator method. I've been coming back to that over and over. Again, Sanger sequencing all possible outputs of the test as a comparator would be hugely expensive. So it's not feasible, and in some cases it would be inadequate, or it may not be the right comparator. If you think back of array CGH or chromosomal microarrays, we've been trying to figure out the right comparator method there too, and we have been coming up with a composite comparator method consisting of maybe FISH, karyotype, other methods, qPCR, microarray consensus can be considered. So there are ways to find some kind of consensus or some kind of best reference method. So this slide gets down to a little bit more specific. If we get down to that, and if we're evaluating a specific NGS-based assay, best current recommendation for assessing accuracy for a targeted assay, or let's think of specific panel of cancer genes, for example, this approach would consist of three parts. First one, sequence clinical samples that represent the intended use population. The second part, sequence procured samples that are adequately representing variation types that are part of the assay. For example, small indels, SNVs, etc., large deletions, whatever is a part of the test. And the third part, sequence some well-characterized reference samples with known consensus sequence deposited in a well-curated public database. And you can assess error rates across different amplicons from that. Of course, there are many other questions that come with that, but it's probably a good start. This slide lists some of the criteria considered for analytical evaluation, somewhat like the QC metrics before one can consider test results are valid in the first place. A lot of these came from Broad and a lot of research and a good work that pioneering work they have done there, and we kind of just lifted that. For example, things to check include transition versus transversion ratio, number of expected calls, allelic fractions, things like sequencing depth for a specific application type, level of coverage necessary to make accurate calls, completeness, uniformity of coverage. These are all things that need to be determined and validated. We do not necessarily expect perfect performance. It is important to understand, though, what the performance is and which regions are problematic to call and which regions can be called perfectly. And we know some regions are difficult to sequence. I think somebody presented today about 10% of the genome has not been sequenced really yet with any of the current technologies. It is reasonable to expect different performance in different regions. Some regions are harder to sequence, some are easier, some cannot maybe be sequenced yet. I want to mention that, a lot of this has been also covered in some of the recent publications that came from about last year or so. And there is a recent newest guideline. Most recent guideline is from ACMG, which was published less than a month ago, July 25th, on clinical lab standards for NGS. So these are for the labs, but a lot of this is a very useful information. So this slide lists very general precision study requirements. These would be applicable to any IVD. So we're talking any IVD, including NGS, because what you see here is very general. Precision studies should assess major sources of variability and include all analytical steps of the assay as applicable. And I will not dwell much more on that. If somebody has questions, we can discuss. So I want to move instead to the clinical performance. Generally, when there are well-established links, such as genotype-phenotype correlations, FDA has been accepted relevant literature, professional societies, recommendations, etc. And we have not been asking test manufacturers to reprove clinical significance and reinvent the wheel, so to speak. However, there are many cases when there is not enough information, and in those cases, we would likely require a clinical study to establish some kind of clinical study, maybe not necessarily a clinical trial, to establish clinical correlation. So the question is, how can this, what has been used so far, how can this apply to NGS tests? I want to start with saying that in the spring of 2012, ACMG issued recommendations on scenarios when to use genomic sequencing and especially whole genome sequencing clinically, and these are listed on this slide. So how can we build on what we have been asking in the past for clinical validation, and then the various recommendations from the societies, like what I've mentioned. So what we have had in the past is we had either, in some cases, relied on published literature, and how can we try to put in place some kind of reasonable interpretation system for high content tests like NGS, and maybe use what's existing in the public domain. So one thing that we have heard from clinical experts during public discussions of cytogenetic arrays that I've mentioned several times is that human and expert interpretation is still critical in these cases. And you've heard that over the last 2 days, if you listen to any of the presentations, how much of the interpretation is manual. So that should definitely be taken into consideration when devising review policy for next-gen sequencing tests. So the interpretation, human expert interpretation. More general, there have been numerous private and public efforts to build what you can call clinical grade databases. And these can be used then, or labs are using them in interpreting all this vast information resulting from NGS. Now, the problem is that a lot of these efforts have, unfortunately, for many very legitimate reasons, been siloed. And there is a need for these databases to talk to each other, and a need to build a unified resource of clinically relevant variants. So a number of work groups are currently trying to push such database for genetic variation, some kind of unified database. And these groups include ACMG, which has been very active in this field, National Human Genome Research Institute, International Collaboration for Clinical Genomics, or ICCG, also very active in both arrays and next-gen sequencing now, and others. I want to mention specifically NCBI's ClinVar, which may be the common database for depositing the data on which clinical interpretation can then be built upon. And many people, this has been live from, ClinVar has been live since April, and many people are depositing data, both clinical labs and researchers. So if successful, we see a clinical grade database as an important part of clinical validation for FDA submissions too. And depending on the specific test and depending on the specific indication, test manufacturers may not need to additionally prove clinical significance beyond what's in the database, but that will be, again, test and indication specific. And I will elaborate some more on database efforts a bit later. But before that, I want to go to the last part of FDA review, and that's test labeling, or package insert, if you will, which according to the labeling regulation, which is listed on this slide, this regulation requires not just instructions how to perform a test. That's not the whole labeling. The labeling, according to this regulation, needs also to list the summary of test performance. Once the test is FDA approved or cleared, we post, as I mentioned before, summary of reviewed performance on the web. Now this slide has two links, on the top to any FDA cleared test, and on the bottom to any FDA approved test, which is when you have to submit a PMA. In those cases, you can access both FDA generated summary of the test performance, but also you can access manufacturer's original labeling. So again, through these links, you can access summarized performance of any FDA cleared or approved test, and all this is publicly displayed. So in summary, so far, what I've described is what we review. And basically, I was talking about evaluation of the test performance and with some specifics of what we are thinking about the NGS test performance. So I was talking, again, both in general and kind of gave you the little 101 and provided some specifics. Now, in the second part of the talk, I want to move to possible ways to regulate these tests, some standardization efforts that are on the way, and collaborative initiatives. Again, before moving to that second part, in this slide, I'm extracting some of the most frequent challenges we encounter. And this is not just with NGS-based tests, but really with evaluating the performance of any novel IVD in general. Many of these challenges will probably look very familiar, and they include finding clinical samples to evaluate the performance, figuring out the best comparator to understand what results the better results of the new test are accurate. And also this, as I mentioned before, with array-CGH, this can include some very constructive ways of putting together some kind of composite reference. And then also there is a lack of knowledge about the clinical validity and significance. Okay, so let's look at all the different parts or modules, if you will, of NGS platforms and tests. And probably many people listening are well aware that many of these modules shown here are still very rapidly changing and developing. And that definitely applies to pre-analytical parts, which is your top left box, and also to data analysis software, which is really rapidly developing. So this is your third box on the bottom box on the left. Excuse me. For example, let's focus on the analysis part. So most platforms use FastQ, as many of you know, for base calling and quality. Now, in many cases, these programs are somewhat different, and they're platform specific. Actually, one FastQ is not the same as the other FastQ. So the most commonly used variant files, on the other hand, seem to be evolving as we speak too. So we're talking VCF, GVCF, genome variation format, or GVF. Some of these may not be the best suitable for clinical needs. Many of them really developed from research and may also work better. Some of these may work better with outputs from some of the platforms than others. So there is a need to standardize at least some of these formats. to allow data exchange between different platforms and software and achieve some level of interoperability. Health Level 7 or HL7 is organization that is involved in development of international healthcare informatics interoperability standards, and there is a group currently working on HL7 healthcare IT data standards for clinical genomics actually. So if we focus, okay, so let's go back to this middle box, what I marked in red. If we focus on sequence generation and detection part, these platforms have actually reached some stability, and some of them are being manufactured under GMPs actually. So we already discussed earlier some possible ways of evaluating performance of NGS sequencers, especially accuracy, but I want to delve a little deeper into some of that, how to now regulate these. So if we build on this previous slide and think about the whole system and all these modules, in general, the way FDA reviews diagnostic tests, it's starting from a sample taken from the patient all the way through pre-analytical parts, instrumentation, and assay software to the result. However, at that meeting that I mentioned 2 years ago, that happened 2 years ago, there were many suggestions that in the case of NGS, we should actually look into some ways of decoupling a platform from the test and perhaps regulate them separately. And I have to say that we are seriously considering some ways to do this. We are trying to create a pathway for maybe getting instruments cleared without a specific indication, and then getting assays approved will be easier on the instrumentation that already has established performance, is manufactured under GMPs, design controls, etc. So we are looking at, and that's what's listed on this slide, at regulating these tests in both ways actually. So classifying just the platform, and we have some possible precedents for that, and the other way would be regulating the whole system or specific assays that are intended for specific indications that can be put on the platforms that are maybe cleared separately. I've mentioned several times the different public meetings and the input from the experts, so we haven't been working in the vacuum. We haven't been trying to figure this out in the vacuum. We have been collaborating for a couple of years now, both within FDA and then across government agencies, with academia, professional societies, with talking to clinical labs and others on a number of standardization efforts to try and leverage various existing efforts and expertise, and to try and help in smoother transition of these new technologies to the clinic, while still assuring the accuracy of the results and also transparent test performance. So what's listed on this slide, there are many efforts actually listed on these slides, and among them, on this slide, and among them, I want to mention several CDC-led initiatives. These have been actually instrumental in developing some of the standardization over the last several years, and they resulted in published recommendations, next steps 2, as well as information about possible reference material GetRM program. Also, CLSI, or Clinical Lab Standards Institute, has been working on developing a consensus guideline for clinical sequencing, which is almost finalized from what I know, and should be also very helpful once available. And I've mentioned already the ACMG guideline, recent ACMG guideline publication, which is also, I understand, publicly available from an ACMG website. I do want to, okay, next slide. I want to highlight actually 3 standardization initiatives. First one, we are working, that we are part of. We are working with NIST to develop human genome reference materials for NGS that will allow both the FDA as well as test and platform developers to understand how their systems work on a well-characterized, consistent material. George Church actually mentioned this material in his talk yesterday, we will be using some of his PGP samples that are well-consented samples for this effort. This collaboration is expected to help standardize high throughput sequencing for clinical applications and facilitate development of safe and effective regulated diagnostic applications. The second initiative I want to mention is a CDC-led effort to address non-standardized software formats for use in the clinic. I was talking about FASTQ and VCF formats and different other formats. So this initiative should help make data coming from various sequencers interchangeable and easier to understand and compare. So this effort is also on the way. And lastly, I want to emphasize that we strongly support the development of a clinical grade database. And in the next few slides, I will try to elaborate on some of the issues encountered for the database specifically. You have seen this same slide earlier, but now I will spend some time over the next few slides focusing on this last part, which is interpretation of the results, and I will talk about the database role and beyond what I already described. Okay, so this unified database that I've mentioned several times now, when we are referring to what you can call clinical grade database that we can also use at FDA, what does this mean? First, there would have to be an appropriate data repository, and I've mentioned before ClinVar as a very good candidate for that, on top of which all the other elements can be built. That data then need to be curated to see where it supports clinical significance. There are some repositories of clinical trial data actually currently in the works by some of the groups. Now, having clinical trial data is nice because there's so much control in generating it. However, while that's great where clinical trial data is available, and it is great to be able to utilize this enormous amount of information that is stored from those trials, we cannot realistically expect to have such controlled studies in most cases. So people are looking at alternative ways to use somewhat less standardized data to learn things about not just test significance, but also things like test-drug interactions. I want to make clear that FDA is not looking at building a database internally. With all the efforts already on the way and all the accumulated expertise in places such as NCBI, for example, and various ACMG and other efforts for germline-related variations, having yet another database effort would not make much sense. So what we are looking at is to help what seem to be the most promising efforts that are either started or they're on their way or starting just now. And what we're looking to is that in that process we get out of these databases the information that we and test developers can use as a part of clinical test evaluation. Some questions to start from include, so where do we start when we are looking, what do we want in the database, right? So these questions may include surveying what may be the gaps in current practices for content or formats of existing or developing databases. We can also look at whether there is some type of curation and how thorough it may be. I was mentioning before manual expert interpretation and curation, and that's still the case. We can compare levels of evidence that FDA would typically accept to the levels that are applied in database curation. It seems very reasonable and plausible there can be a stepwise approach to both quote, whatever is the raw data collection as well as levels of evidence. At the bottom of this slide, I listed some of the current database efforts underway and who are the main drivers behind those. for different types of variations. So germline, somatic, microorganisms. I didn't list the drivers there, so that will be NCBI. I think also EBI and definitely FDA's CIFSAN, which is Center for Foods, are working together there. Some options that are being discussed are whether it is more practical and realistic to have one centralized database, or perhaps federated approach would be more feasible and maybe better suited to diverse needs and laws if building an international database with a range of stakeholders that can use it for their specific purpose. If the approach is some kind of federated database, there would presumably be some kind of apps on top of these various separate databases that would interconnect them and be able to pull the data that someone may need for their specific clinical use. Another question often discussed is the need for different evidence levels and to store not just clearly clinically validated data, because there's so much more to learn from additional data that may or may not eventually ascend to some type of clinical actionable level. Additionally, having a feedback loop to deposit or correct the information we learned more about since first placed in the database, or when some expert group curates it, is very important and should be built in. I mentioned already the need to not restrict data to only clinically actionable. This need for not restricting is actually applicable through all different levels of the possible database, not just having different rigor of curation. It should be, however, very clear what is in the database, what is at each level if you're having different levels, and how validated and curated it is. There seems to be a general agreement that both research and clinical data can be deposited, from what I know, as long as it's noted or delineated where the data comes from and with the understanding of the level of validation and controls. Next question that NCBI has been wrestling a lot is the question of some type of unified data formats. I've mentioned that twice already. The naming conventions, similar issues, to be able to analyze together data coming from various disparate sources. There have been some successful attempts, actually, and I think ASCO pilot database may be one, to create platforms or applications that can take in very different non-standardized data types and consolidate them. But this was not, any of these pilots, from what I know, were not with genomic test data. But this possibility of using non-standardized data may be very important, actually, since labs generating and submitting the information may not have additional time or resources, at least at the beginning, to convert their data to some kind of standardized format. Additional issues people are working on include privacy issues, especially if collecting data and information coming from different entities, different countries, different that would have different laws, so that's important. And lastly, I want to mention options like cloud storage versus local servers, and what should be stored, where these are increasingly considered. So where do we go from here? Well, we are working towards hopefully having some FDA-cleared sequencing platforms and tests in the near future. It also might be helpful. We are thinking to publish some kind of points to consider document. So what points to consider when evaluating next-gen sequencing-based tests for FDA submissions. Many people have told us that would be helpful. I want to say that we are very interested in feedback from other experts in the field, and we are very open to hearing suggestions and criticisms. If you think we've done something right, we wouldn't mind hearing that either. But definitely we want to know about holes in our approach, and we would like to work in overcoming those. I want to end with also saying that if someone is considering submitting a high content test for FDA clearance, we encourage you to contact FDA early in the process. And with that, I want to thank you very much for listening. Thank you very much for joining. I would be happy to take any questions, and on this slide also I'm providing my email address. So if I don't know the answers to your questions, or if you want to contact someone at FDA directly and you don't know who to contact, please feel free to contact me and I can direct you to the right person. And I see there are some questions coming in. And also, I think I need to end with this slide. If you need CME credit, go to this web page. Okay, let's look at the Q&As now. That's okay. No questions available for display, even though there were incoming questions. Okay. Well, so I think I can maybe wait for a second, but I do want to say again, I want to come back to this slide. If you don't have questions now, or if you think of some later, or if you don't want to ask your question publicly for whatever reason, please feel free to contact me. And if I don't know the answers, again, I will make sure to direct you to the right person, because we are very open to both feedback and also to giving you feedback on whatever your test is. So if there is no questions, if it's all clear, I think I will end the broadcast here.